Hey everybody, welcome to chapter 10. This chapter covers patterns of inheritance. So last week or last chapter, what we discussed was chapter 9, which was regarding primarily meiosis. And meiosis was the formation of gametes, which are sperm and egg. So this chapter goes on to extend from what we learned in chapter 9 to how the creation of sperm and egg and the processes that went on during that chapter affect the patterns of inheritance, because of course we get our genetics from our parents, and that genetic information is passed down via the sperm and egg that are created via meiosis. Okay, so let's first start off with defining a couple of terms. Those terms are heredity and genetics. Heredity, by definition, is the transmission of traits from one generation to the next. And when I say traits, I'm talking things like eye color, hair color, your skin complexion, the shape of your eyes, the ability to taste PTC on a strip of paper, the shape of your hemoglobin protein. So when I say traits, it's not just physically the things that you can see macroscopically, which means with the human eye, but also things on a microscopic level and a molecular level as well. What proteins you have, the shapes that they are, etc. It all is a result of the transmission of genes, which is heredity. The next term is genetics, which is the study of heredity. The type of heredity that we're going to discuss in this chapter is called Mendelian inheritance patterns, which stems from the person that first came up with this idea, whose name was Gregor Mendel. But I want you to note right here that not all characteristics are passed down via Mendelian genetics. Mendelian genetics is a very simplistic type of inheritance pattern. It's a great starting point for us to start thinking about inheritance patterns, but do know that not all traits are actually passed down via Mendelian genetics. So I mentioned Gregor Mendel. He was the one that came up with the ideas behind our modern day interpretations of Mendelian inheritance patterns. But before Mendel, people were pretty accepting of the concept of blending patterns of inheritance. And so what that means is that the parents of contrasting appearance that look different from one another produce offspring that are a blend of the parents. So for example, we have these flowers over here. This is demonstrating a red flower mating, that's what the X means, with a white flower, and their offspring are listed here, and they're pink. So if you are familiar with their colors, which I'm sure y'all all are, if you mix red paint with white paint, you're going to get pink. And so this is the theory of the blending concepts of inheritance. However, it was known that this type of model does not explain variations of blending. So, for example, let's say we have a blonde-headed mother and a red-headed father. Based on the theory of blending concept, their offspring should always be a mix, like a strawberry blonde. However, that's not always the case. So this type of inheritance pattern does not explain those variations. And so Mendel completed experiments to counter this findings, and his results did counter the findings that his findings were in contrast with the blending concept, essentially. So because of the things that he came up with via his experimentation, Gregor Mendel is considered the father of modern day genetics. All right, so let's discuss who Gregor Mendel was. He was an Austrian monk. He worked with garden pea plants. Garden peas, that's going to be important. And this is around the 1860s, so, a good while ago. And back when he began his work, most people acknowledged that both sexes contributed equally to a new individual, but they just weren't really sure on the details of how. They would, like I said in the previous slide, they weren't able to account for variations among members of families over many generations. They had no knowledge back then of what cells were, or even, of course, what then chromosomes are, which is so, it's very impressive what you're going to come to learn that he established in this PowerPoint. These things that he figured out were done so with no knowledge of cells, no knowledge of chromosomes. They didn't know what DNA was. They didn't even have microscopes at this point. Microscopes came shortly after this, relatively to the grand scheme of things. But at this point, he did not have a microscope. So what he did have in his toolbox was his ability to understand the processes behind statistical analysis, and he had garden pea plants at his disposal. So what he did was he gathered experimental data from garden pea plant breeding and looked at that mathematical data and drew conclusions from it. So why the garden pea? So this is a typical garden pea right here. You see it has flowers. Now, the reason why he chose this particular species of plant as his experimental model, we call these model organisms. They help model how things happen in humans. Even though this is a plant, it still is useful in modeling human genetics. But the first reason why he chose garden peas is because they're easy to cultivate, which means they're easy to grow. So that means we can grow a lot of them without putting in a whole lot of work and monetary efforts as well. Think about the difference in the cost of growing a flower versus how much it would cost to raise lab rats or pigs. A lot more expensive when it comes to animal models. So here we have garden peas, which are very easy and cost effective when considering cultivation. They also have a short generation time, and what that means is that they develop really fast. So if you think of, the generation time of a human, you're looking at around 20-ish years. So that's the time from birth to the time where that individual gives birth to a new generation. So again, 20 years is a very long time. We need something that's going to have a short generation time so we can do lots of experimentations, have lots of samples to observe good statistical analysis. Remember, back in chapter 1, we talked about statistical analysis and how the more samples that we have, the better. The more closer we are to true, accurate data that is not subject to chance. Okay, so short generation time, they grow within, a few months. They're also able to produce true breeding plants. And this right here, that word is going to be very important later. Right now, I want you to know that true breeding plants have offspring that always look like the parents. So if this particular pea plant here was true breeding and it was bred with another pea plant, it would have offspring that all looked like it. So for example, if we're talking about flower color, if it was true breeding for that trait, then all of its offspring would produce purple flowers. And you'll see why on a molecular level or genetic level later on in this PowerPoint. But right now, know that true breeding produces plants that always look like the parents for any particular trait. Another important characteristics of garden peas that made them a great tool for Mendel was that they normally self-pollinate. Okay, so if any of y'all are familiar with gardening, or heck, even if you're not familiar with it, you've probably heard of pollination, right? If not, all it means is that sperm and egg of plants meet. So normally these plants are self-pollinating, which means the male components of the plant and the female component of plants are on the same plant. And they are able to self-pollinate then by itself. The male flowers will produce what we call pollen. Y'all know what pollen is. That's kind of like plant sperm, quote unquote, if you will. And that pollen will pollinate a female flower on the same plant, or of other plants if there are other plants nearby it. So normally they are self-pollinating. They are capable of doing that, but they can also be cross-pollinated by hand. This is called manual pollination. And manual pollination is where you physically take pollen, which is plant sperm, from one plant and brush it onto the female organs of plant number 2. That way, you're able to control who the parents are and then see what the offspring look like. Okay, so again, Mendel used the garden pea. This is the scientific name for the garden pea, Pisum sativa. He chose true breeding varieties where the offspring looked like the parents, and he kept careful records. He kept very careful records of very large numbers of experiments. So like I said in the beginning, he had a great understanding of mathematical laws of probability, which helped him with his statistical analysis and interpretation of the results that he got. And based on his finding, he came up with the particulate theory of inheritance. Now, I want you to think about what that means. Particulate. What does it mean when I say a particulate? If there's particulates in the air, what am I talking about? Particulates mean small things. So his particulate theory of inheritance is based on the existence of minute particles, which we now know as genes, because again, back then they didn't know what chromosomes and genes and DNA and cells were. Now we do. But his theory was based on the existence of these minute particles that he didn't know what it was being passed down from generation to generation. And this is our modern day gene theory. All right, so this is a summary of Mendel's work with his garden peas, and it elaborates on the different characteristics that he observed. And I'm seeing right here, I've got a little bit of this is hanging off here, so I'm going to escape, discard my annotations. Uh-oh. Not wanting to do that. There we go. Okay, so y'all can see that a little bit better now. Sorry. All right, so this is a picture of Mendel here observing one of his pea plants, and this shows some of the characteristics that he looked at and experimented on with these garden peas. So, for example, stem length. Some plants had really tall lengths and were very tall plants, and some were short. He observed pod shape. So you can see this pod here is very inflated, whereas this one is constricted around the peas that are inside of it. He observed seed shape. There's 2 varieties. There's round and wrinkled. Color. There's 2 varieties, yellow and green. Flower position. Some of them had axial flowers, and the other option was terminal flowers where they're on the end. Flower color. They were either purple or white, and pod color, green or yellow. So you'll also note over here, it says characteristics that are dominant are up top and characteristics that are recessive are on the bottom. And so just kind of keep a mental note of those terms, dominant and recessive, because it will be important, but I don't want to talk about those just yet. We'll get to it later on in this PowerPoint. But these are the different characteristics. Notice they have 2 varieties per characteristic that he observed when working with his garden peas. Okay, so let's look at the garden pea anatomy when it comes to their reproductive system so we can better understand manual pollination. So here is the flower of a garden pea. If you look here, we see the stuff in yellow, these filaments here, long strands with these little balls at the top. Those are called anthers. And the anthers of plants produce pollen. And the pollen grains are the sperm of the plant itself. So when that pollen grain comes in contact with the female organ of the plant, which is the stigma right here in green, it travels down the stigma and into the ovary of the plant here, where it can be fertilizing, or where it can fertilize the egg, which is located in the ovary of this plant, that will then develop into seeds that can then go on to produce a new plant, or, aka its offspring. Okay, so again, the yellow stuff here is the male component of the flower that produces pollen, which is plant sperm. And that plant sperm can travel to the stigma of the female reproductive system of the flower and down into the ovary to fertilize the flower's egg, which then will produce a seed, and the seed will produce a new plant. Okay, so now that's self-pollination. So what Mendel did is he cut away the anthers of the flowers. So if the anthers are cut away, there's no pollen that is going to be able to be passed to the female component, because he's separating them. So the plant never meets, or excuse me, the sperm never meets the egg. What he did instead of allowing for self-pollination is he cut away the anthers. He physically took a paintbrush and rubbed it onto the anthers that he cut off, and then used that brush to apply the pollen to another plant that had also had its anthers removed. So that way he could manually determine which plant would create offspring. And so he would observe the offspring of those plants and be able to make assumptions after gaining lots of data, lots of samples of doing this. He would be able to observe the offspring's characteristics based on whatever plants he manually decided would pollinate each other. So this is kind of like, I'll give you an example in animals, which might be easier to wrap your head around. It's kind of in a similar sense. It's sort of like artificial insemination. So, for example, if breeding horses is a common place where artificial insemination would occur. If you have two horses and you want to breed them together because you're hoping to get characteristics from horse A and horse B, you would use artificial insemination to do that, unless they were willingly going to breed together without doing artificial, which is possible. Kind of like breeding dogs. You want two dogs to breed together because you hope that the offspring will have characteristics from both of those parents. You put them together at the right times and hope that they breed together. This way that Mendel did it just is a manual process, artificial insemination, that you basically are taking the sperm of organism number 1 and inseminating organism number 2 with it. Okay. So the first types of experiments that Mendel did were based on one-trait inheritance patterns. So what I mean by that is he crossbred or hybridized, that's another term for that, organisms that only differed in one trait. We call that a monohybrid cross. Okay, so Mendel performed crossbreeding experiments between two true breeding individuals, and again, true breeding means they always look like their offspring always look like the parents when they're bred with another individual that looks like it. We call that hybridization because we're crossing two individuals. And they only differed in one trait. We call that a monohybrid cross because mono means one. So, for example, he would have manually crossed two pea plants that had all of the same traits except for flower color, for example. So that means that they were both tall plants that were inflated with their pods. They had round seeds. The seed color was yellow. They had axial flower positions, and they had green pods. But one plant had purple flowers and the other one had white. So all of the characteristics were the same except for one thing. That is a monohybrid cross, and you need to know that definition. Okay, so When he did his experiments, he allowed for the parents, the parental parents that he chose to be called the P generation, and P actually stands for parental, right? So P, parental, they're the parents. They're the very first generation. The offspring that they produced was called the F1 generation. And that's because F stands for filial. And that stands for a Latin word that means offspring. So the first filial generation is the first offspring. The second generation, which would be the offspring of the first filial generation, was called the F2 generation, or the second filial generation. And what he did in the parental generation is he crossed green potted plants with yellow potted plants. They were the same in all other characteristics except for pod color. And what he saw in the F1 generation is that all the plants had green pods. So at first it appeared that the yellow podded trait was just gone. But he went a step further. He allowed for self-pollination of the F1 generation. So the F1 generation, these offspring, were allowed to pollinate one another and produce the second generation. And in that second generation, yellow pods reappeared in a 3-to-1 ratio. He did the same type of experiment with all those different traits that we discussed previously. Flower color, flower location, pod shape, seed color, etc. All those other characteristics back up, all of these different characteristics, and he saw the same thing every time. If this was a purple flowered plant and this was a white flowered plant, all of them would have purple flowers, which is the dominant trait, in the F1 generation, and the white flower would reappear in the second generation in a 3-to-1 ratio, where for every 3 purple flowers, there is 1 white flower. Just like there's 3 green pods in this experiment and 1 yellow, a 3-to-1 we call that a phenotypic ratio, and we'll get to that definition in just a minute. So, and I know there's some things on this chart, like these G's and little g's and big G's, that you may not know what all that means yet. Don't worry about it. I'm going to come back to it, but I want you to get the gist of this before we go into the details. Okay, so again, the first generation, the very initial parents, we call that the parental generation. Their offspring is called the F1 generation, and all of them have the dominant trait. And then in the third generation, which is the offspring of the F1 generation, we have a reappearance of the recessive or non-dominant phenotype or characteristic trait. So green is still in the majority. For every 3 dominant characteristics, there is 1 recessive, but it reappears in a 3-to-1 ratio. That 3-to-1 ratio, super important. Okay, and here's why. So Mendel, like you know, was very good at math. He was good at statistics, and he was well versed in probabilities. And he reasoned that that 3-to-1 ratio was not a coincidence, considering it happened every single time, and that a 3-to-1 ratio would only be possible if the following conditions were true. And those conditions are that the F1 parents must have contained 2 copies of the heritable factors. He called those unit factors back then, but nowadays we call these genes or alleles. Remember, alleles from last chapter, by definition, are variations of the same gene. For example, if we're talking about people, they would have 2 genes for eye color. But those genes might not be identical. So they could have 1 blue-eyed gene and 1 brown-eyed gene. And we call those alleles because they vary. The second point that he reasoned had to be possible is there has to be a dominant allele and one recessive allele. And dominant and recessive are important in Mendelian genetics. And all it means is that a dominant allele will mask a recessive allele. So if you have a parent that has blue eyes and a parent that has brown eyes, like I do, I have brown eyes. So that means that I either have two dominant alleles, or excuse me, not two dominant alleles, that I must have a dominant allele and a recessive allele. Because I have brown eyes, therefore I must have gotten a dominant brown-eyed allele from my mom, and my dad has blue eyes, so I must have gotten a recessive allele from him because there's no other options there. All right, we'll get in more about that and do some practice with it a few slides down. But there must be a dominant allele and a recessive allele, and a dominant allele will mask a recessive one. He also reasoned that these factors, or genes, that we now know are genes, separated when gametes were formed, when sperm and egg were made. And each one of those sperm and egg carry a single copy for each factor. And then when they meet or fuse during fertilization, they restore having two factors. And then lastly, random fusion of all possible gametes occurred at fertilization. Okay, and so based on his reasoning, he came up with his laws of inheritance. The first law was the law of segregation. And the law of segregation states the following. And I want you to think, what does it mean to segregate? Segregate means to separate. So the law of segregation states that each individual have two factors or alleles for each trait. The factors segregate or separate during the formation of gametes. Remember, in meiosis, we had division of genetic material into sperm and egg in such a way that each gamete only contained one factor for each of a pair of factors. All right, so remember, meiosis started off with a diploid cell, and after the first round of division, which was a reductive division, became haploid cells, which reduced the chromosome number in half. Then, after the second round of division, the sister chromatids separated, creating 4 haploid cells that have half of the genetic information that the original cell did. So this was meiosis 1, meiosis 2, resulting in 4 sperm or egg with half the amount of DNA as the original parental cell. All right, so because it has half, and the way that the processes of meiosis occurred, it winds up being so that each gamete contains one gene for each pair of factors. And then fertilization gives the new individual 2 factors for each trait. So here would be an example of a pair of homologous chromosomes. Remember, homologous chromosomes come in pairs. We have 2 for each type of chromosome. In humans, we have 23 pairs. 1 of each pair came from mom, and 1 of each pair came from dad. So here we have a blue chromosome representing the paternal chromosome, came from dad, the male, and then the red one came from mom. And they each have a gene, which is a location on a chromosome, that codes for flower color. And as you can see, the flower colors here are not the same. These are alleles. They're variations of the same gene. All right, so this would have been carried in dad's sperm, and this one would have been carried in mom's egg. And upon fertilization, we have a complete set of 2 factors for each trait. Okay, so the modern interpretation of Mendel's work is called the chromosomal theory of inheritance, or gene theory. The chromosomes are carriers of genetic information and is passed down via genes. And these traits that we have are controlled by whatever genes that we get upon being created, like upon fusion of sperm and egg. All right, so the traits are controlled by genes that occur on homologous pairs of chromosomes at particular gene loci, or a gene locus, singular. And each homolog holds one copy of each gene pair. And meiosis explains Mendel's law of segregation and why only one gene for each trait is in each sperm and egg. All right, so if you need to review meiosis, please do. But again, if you'll recall, I'll draw some chromosomes in here. All right, I'm just going to draw. This is a duplicated, this says after interphase. This is a duplicated set of chromosomes of a homologous pair entering into meiosis 1. Right? After meiosis 1, our homologous chromosomes separate. The cell was diploid, these will be haploid. Then after meiosis 2, the sister chromatids separate, resulting in 4 haploid gametes. And then sperm plus egg will restore the diploid number. Here's my zygote. But again, you saw, just like in we had separation, segregation of the genetic material into two newly forming cells. And so that is Mendel's law of segregation. Those chromosomes separated, those homologous pairs separated, which ultimately resulted in each sperm/egg getting one copy of each trait. So this one, the sperm, had a chromosome that carried purple flowers, and this one carried white flowers. We would then have a diploid zygote, which had a purple flower gene and a white flower gene. I'm just write P/W for short because I'm running out of space. Okay, alleles we've already mentioned. I'll mention it one more time. They're alterations of a gene. They're alternative forms. They're variations. White flower gene versus purple flower gene. Blue-eyed gene versus brown-eyed gene. Still codes for eye color, just not the same color. And again, dominant alleles mask the expression of a recessive allele. And something we haven't mentioned yet that you need to know, which will kind of make some of this information come full circle, I hope, is that the dominant alleles are specified by a capital letter, and recessive alleles are specified by a lowercase letter. And alleles occur on homologous chromosomes at a particular location called the gene locus. We've said a few times now. All right, so the combination of alleles that you get determines what traits you have. We call the combinations of alleles that you have a genotype. Genotype is the type of genes that you get. So you could be homozygous. Here's some new terms, lots of new terms here. Flashcards would be good for these. Homo means same. So if you're homozygous, you have two identical alleles. A heterozygous individual has two different alleles. Now, there's two options when you're homozygous for a trait. You can be homozygous dominant or homozygous recessive. So if I were using G's for eye color, for example, and brown is dominant, then I would say that if you're a homozygous dominant individual, your genotype would be big G, big G. If you're a homozygous recessive, you would be little g, little g. If you are heterozygous, you would be big G, little g. And I could use any letter here. If I ever ask you a question or if you've ever given an example, it will always let you know what the letter that we're using is. I could have just as easily said we're using A's. And if that's the case, a homozygous dominant individual would be big A, big A. Homozygous recessive would be little a, little a. And a heterozygote would be big A, little a. Now, depending on what combination of genes or alleles that you have, it will determine your phenotype. And the phenotype is the physical appearance. So When I say physical appearance, I do mean the macroscopic qualities like your eye shape and eye color and the color of your hair and your skin and your blah, 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 and how tall you are. But it also is talking about how you look microscopically. What is the shape of your hemoglobin protein? What is the your normal level of your thyroid hormone? What's your reference range for you? All right, so literally everything is determined by your genotype, and we call that physical appearance the phenotype, even though we may not technically be able to see it because it's too small. It's the physical trait. All right, so here's a table showing you genotype versus phenotype and the allele combinations that are appropriate for each one in regards to skin pigmentation. So we're using A's. Again, we could have just used B's, C's, D's, or Z's. It doesn't matter. But there are 3 possible genotypes. You can be a homozygous dominant. And if that is the case, then you have 2 big A's. And in this circumstance, you would have normal pigmentation. In a heterozygous individual, you have 1 dominant allele and 1 recessive allele, and also would still have normal pigmentation. Because this normal allele masks the recessive allele, so you don't see it because the dominant allele masks it. And then lastly, the third possible combination of alleles is little a, little a. You have 2 recessive alleles here, so you are considered homozygous recessive. You have 2 alleles that are the same. Homozygous, homo means same, and they're both recessive. And under this type of genotype, it results in albinism. Albinism is the lacking of skin pigmentation. It's the absence of melanin, which is actually the protein that produces that pigmentation of your skin complexion. All right. The next item up for discussion is the Punnett square. Punnett squares are a useful tool that helped us predict genotypes, phenotypes, and their probabilities that will occur from a particular cross or breeding experiment. So the way that we set up a Punnett square is by drawing a square and dividing it into 4 quadrants. All right, so you see this square here. I'll draw my own as well. 4 quadrants. We will list all possible sperm genotypes up on one side and the egg genotype on the other side. So in this case, we are crossing a heterozygous female with a heterozygous male. Hence why they put big G, little g on both sides. Male and female. Then they place all of the possible zygote genotypes within these squares. So these squares, these 4 squares, represent all of the possible offspring genotypes that could be created from this cross of this female with this male. The way they do that is by dragging the letters down and across appropriately. And the best way for me to demonstrate that is just to show you. So in this box here, we'll drag down big G, and from over here, we'll cross to, or we'll put big G here too. So here we have big G, big G. Then we'll take this big G and move it over here. Big G and this little g from the female. Same thing, move this down, big G, move this across, little g. And then move this little g down and move this little g across. So this shows you then that there are 3 different possibilities when it comes to genotypes of the offspring. They can be big G, which is homozygous dominant. They can be little g, which is homozygous recessive. Or they can be heterozygotes, here and here. Now, furthermore, this tells us more information than just what's possible. It also helps us with understanding the probabilities of these events occurring. So, this is saying, because again, you divided this into a quadrant. If you divide 100 divided by 4, you get 25. So there's a 25% chance that the offspring of this cross will be a homozygous dominant. There's a 25% chance that they will be homozygous recessive. And there's a 50% chance, because 25% plus 25% is 50%, there's a 50% chance that the individual will be heterozygous. Those are called the probabilities of genotypes. Now, the probabilities of phenotypes is different. So genotype, again, is the combination of alleles, the combination of capital G's and little g's that we got in each one. The phenotypic ratios are what organisms are going to look like. So even though this one is a heterozygote, and so is this one, they're going to look the same as the homozygous dominant individual, because the dominant allele in the heterozygotes are going to mask that of the recessive allele. So all three of these are going to have green pods, where this one is going to have yellow pods because yellow is recessive characteristic. So there is a 3-to-1 phenotypic ratio here. We have a 3-to-1 phenotypic ratio here. 3 green pods for every 1 yellow pod. If we were doing this for genotype, we would say there's a 1-to-2-to-1 ratio. That's a genotypic ratio, which is the ratio of your homozygous dominant to your heterozygote to your homozygous recessive. 1-to-2-to-1. All right, so this image here on the right shows you an image you've already seen. It was a Punnett square. You just didn't know it was called that when I first showed it to you. And now that we've elaborated on all of the different parts of the contents of this Punnett square, we'll review it one last time. So the P generation is true breeding. True breeding. So what that means, now that you know what dominant recessive is that these plants must have either 2 dominant or 2 recessive alleles. And you can see, in fact, that they do. This plant has 2 dominant alleles. This plant has 2 recessive alleles. So they are true breeding. If you were to breed this yellow podded plant with a plant that also had yellow pods, all of their offspring would have yellow pods 100% of the time. If you were to breed this green pod plant with a green podded plant, then all of the offspring would have green pods 100% of the time. That's what it means to be true breeding. Now that you know those definitions. They have 2 dominant or 2 recessive alleles, and thus when they are bred with an organism that looks like it, it will always produce offspring that looks like the parents. So the next thing on here now that you should understand is that the gametes. These demonstrate the gametes in the P generation, the sperm or the egg. Well, because this green potted plant has 2 big G's, the only type of gamete that it can offer to its offspring is a big G. Likewise, the only gamete that can be, or the only gene that can be given from our yellow potted plant is a recessive allele, little g. So that means that all of the plants that are a result of this cross will have a big G from the green potted plant and a little g from the yellow potted plant, and will be heterozygous. Big G, little g. And because big G, which is the allele for green pods, is dominant and the one for yellow pods is recessive, the green pod will show. So we will have 100% green potted plants in our F1 generation. Now, if the F1 generation is allowed to self-pollinate, that means that we will have a female that is heterozygous, big G, little g, and a male that is heterozygous, big G, little g. And if we were to fill in the chart, so big G, big G, big G, little g, little g, big G, little g, little g, then we will see that 3/4, 1, 2, 3 out of 4, or a 3-to-1 ratio of green to yellow phenotypes. And again, it would be a 1 to 2-to-1 if we were talking about genotypic ratios. Another thing I want to note here that you'll notice in the textbook is that you'll see that the capital G always comes first if it's available, and that's just a formality. So it's kind of like grammar. Big G, little g is the same thing as saying little g, big G. So if you see me do it differently than what you will see in the textbook, then don't get confused. It means the same thing. It's the same combination. It's just in a different order, and the order doesn't really matter. Again, it's just a formality like grammar. But especially when we get to dihybrid crosses towards the end, whenever I do them, I don't always put my big G's first because it makes me, my brain just doesn't work that way. And some of y'all's might work the same way mine does. So just keep that in mind. It means the same thing. All right, so let's do an example. Here we have the P generation. We have a tall plant and a short plant. They are true breeding because they are homozygous. Homozygous dominant, homozygous recessive. Here's an allele key. It shows you that. Big T is tall plants, little t is short plants. It also gives you a phenotypic key. If it's yellow shaded like this one, it means the plant is tall. If it's like the greenish bluish shade, then it's short. So the gametes that they are possible of giving to offspring are as follows. This one can only give a big T. This one can only give a little t. So those are our P gametes. So that means in our F1 generation, we will have 100% of the time a heterozygous combination of alleles, big T, little t. That means that these plants will all be tall or short. You tell me. They will all be tall because the tall allele masks that of the short allele. There's no in between, tall or short. So now what about the F2 generation? The F2 generation will be where we have two organisms from the F1 generation that will breed together. So we will have, here's our F1 generation, we will have a female that will be big T, little t, and a male that will also be big T, little t. So what will go in this first box in the top left-hand corner? If you said big T, then you're correct. What about this box? If you said big T, little t, then you're correct. What about this one? If you said big T, little t, then you're correct. And lastly, what about this one? If you said little t, then you are correct. And so lastly, let's point out one more time our phenotypic ratios. We have 3 tall plants because they have at least one dominant allele. 3 tall and 1 short. That's a 3-to-1 ratio. So I want to ask you again, what would be the genotypic ratio? Because this was phenotype. Phenotype is how it looks. The genotypic ratio is the ratio of the genotypes, starting with homozygous dominant. There's 1, there's 2 heterozygotes, and 1 homozygous recessive. All right, here's one more example that's kind of fun because it applies to humans. So some people have attached earlobes versus unattached earlobes. The person, let's see, right here is an example of a attached earlobe. So you can see they don't really have a very large indentation right here as this person does. So you look in the mirror and kind of tell if you have unattached or attached earlobes. So this is a trait that applies to Mendelian inheritance patterns. So we're going to have two parents here that are both heterozygotes. So the male has a big E, little e, female, big E, little e. And if you fill in the chart, we'll see a 3-to-1 ratio of phenotypes and a 1 to 2 to 1 ratio of genotypes. All right, so one thing you might have questioned at this point is, well, how did Mendel know that the original parents in the P generation were true breeding? Because if you have a green potted plant, it could be a homozygous dominant, or it also could be a heterozygote, which would affect the tallies of the numbers of green potted versus yellow potted plants in the offspring. So what he used to ensure that the original parents were true breeding was called a test cross. A test cross. A test cross is used to determine whether or not an individual with a dominant trait has two dominant factors for that particular trait, meaning they're homozygous dominant, or if they're heterozygotes. We don't need to do that for the recessive trait, because if it has a yellow pod, if it has the recessive physical trait, then it has to have two recessive alleles, because if it had one of each, then the dominant phenotype would be seen. So this is only used for organisms that have the dominant phenotype. So Mendel crossed his F1 generation, his green potted plants that had the dominant characteristic, with true breeding yellow pods. And again, yellow is recessive, so they have to be little g, little g. So there's two possibilities here. If his F1 generation was only has one dominant factor, meaning they're a heterozygote, then the results among the offspring would be a 1-to-1 ratio. If the parent with the dominant phenotype in the F1 generation has two dominant factors, meaning they are big G, homozygous dominant, then all of the offspring would have a dominant phenotype. And so you can see that displayed here. This is option 1. They have one dominant factor and one recessive factor. That means that they are big G, little g. And then the yellow potted plant, of course, has to be little g, little g. And there would be a 1-to-1 ratio. We can do the Punnett square if you like over here. Little g, big G, little g. %50 of the time they would be yellow, and 50% of the time they would be green. That's a 1-to-1 ratio. All right. Option 2. If the parent has two dominant alleles, they're a homozygous dominant organism, and they're crossed with a yellow potted plant, then all of the offspring would be heterozygous, and therefore all have green pods. Okay. Now let's move into a dihybrid cross. A dihybrid cross uses true breeding plants, again, that differ in two traits instead of one trait. So remember previously we talked about monohybrid crosses, mono meaning one. Now we have dihybrid, which means two. So he would cross, for example, tall plants that had green pods, that had a genotype of big T, little, or big G, which he knew because he performed a test cross, with short plants with yellow pods. He did not need to do a test cross for that, because if they demonstrate the recessive traits, then they have to be homozygous for it. All right, so we have big T, big T, big G, big G, which means tall, and green pods with little t, which means short, little g, with yellow pods. So when he performed this cross, the F1 generation had tall plants with green pods. Those are both dominant characteristics. He then allowed for that F1 generation to self-pollinate and observed the phenotypes in the F2 generation. And this is what he saw. This here is what he saw. But before he performed this experiment, he knew that there would be two possible results for the F2 generation. So the two possibilities were that if the dominant factors always go into the gametes together during meiosis, then the F2 generation will have only two phenotypes, which means they would all have tall plants with green pods or short plants with yellow pods. However, if the four factors segregate into gametes independently, four phenotypes would result, meaning that the genes for tall and the genes for height and pod color are either going to be separated into gametes together or independently, meaning on separate chromosomes ultimately. So if that's the case, then there would be four phenotypes that would result from this cross. And that's what he observed, is four different phenotypes. He had tall plants with green pods. He had tall plants with yellow pods. He had short plants with green pods and short plants with yellow pods. That's four different phenotypic possibilities. And based on that demonstration in this experiment, he determined that these four factors, these genes that control plant height and pod color, segregate during meiosis independently of one another. Otherwise, we would have had tall plants that always had green pods or short plants that always had yellow pods. And based on that observation, he created his second law of heredity, which is the law of independent assortment. So what this law states is that each pair of factors segregates or assorts independently of the other pairs. So there is all possible combinations of factors that can occur in the gametes. And as a result, when all possible sperm have an opportunity to fertilize all possible eggs, the expected phenotypic ratio is always 9 to 3 to 3 to 1 in a dihybrid cross. And you need to know that when we have the F2 generation is going to have a 9 to 3 to 3 to 1 ratio in a dihybrid cross. Okay, so in summary, Mendel's laws, there's two of them. The law of segregation, which stated that every single gamete contains one allele for each trait because the homologous pairs separate during meiotic divisions. Number two, the law of independent assortment is a statement that says collectively the gametes have all possible combinations of alleles. Because in meiosis, during metaphase, or metaphase 1 specifically, the homologous chromosomes line up independently of one another. So what that means, here's before meiosis 1, we see I have our homologous chromosomes. Here's homologous set A, here's homologous set B. They duplicate to form X's during the S phase of interphase. And during meiosis 1 in metaphase, when they line up at the metaphase plate or the equator of the cell, there's two possibilities. They can line up like this or they can line up like that, which means we can have the maternal chromosome and then a paternal chromosome. And then on the opposite side, we have a paternal chromosome and a maternal chromosome. Or they can line up this way, where both of the chromosomes are on the right side for dad, and on the left we have moms. Now imagine how complex this gets, y'all, because remember, humans have 23 pairs of chromosomes. This is just showing you 2 homologous pairs for simplicity's sake. But again, they can line up like this, or they can line up like that. And depending on how they line up will result in different gametes. All right, so if they line up like this, all of the dad's chromosomes will be in cell 1, all of mom's will be in cell 2. So after meiosis 2, here's our 4 cells that are possible. We would have this one being little a, big B, little a and big B, because this half, of course, went into that cell, and this half went into that cell. Again, review mitosis and meiosis if you need to. Likewise, over here, big A and little b would have migrated into this cell, whereas little b and big A, this half, would have went into that cell. All right, so that is the possibility if they lined up this way. However, if they lined up like this, then we would have different possibilities at the end. All right, so these 4 go with this possibility. We have these 2 homologs going to this cell, these 2 that went to that cell after meiosis 1, and then after meiosis 2, these sister chromatids went there. These sister chromatids, big B, big A, went here. Little a, little b, little a, little b. All right, so again, this is the law of independent assortment. They can line up at the metaphase plate in either way. All right, so now when completing a dihybrid cross, it's a lot more difficult than it is for a monohybrid cross due to the sheer increase in the amount of stuff you have to fill in here. All right, so the hardest part of it, though, is trying to determine all the gamete combinations that are possible that you need to put up at the top for the eggs and here for the sperm. All right, so the best way to do that is the foil method of the F1 generation. Okay, so I'll start at the top here. The P generation, we're a homozygous dominant and a homozygous recessive individual for both traits. In the P gametes, the things that they are possibly able to give is a big T and a big G, because there's no other possibilities. Likewise here, they had to give a little t and a little g. So that's going to result in the F1 generation being heterozygous for both traits, big T, big G, little g. Now, using the foil method, we determine what goes here, 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 and here. And we'll get back to everything else that's in this chart in just a second. But FOIL stands for first, outside, inside, and last. So first means the first allele of each trait. So using the genotype, we're using A's and B's for the example. The FOIL method says first would be the first allele for each trait. So for trait number 1, we have big A. For trait number 2, we have big B. So that's the first possible gamete. For outside, it's the outside allele for each trait. So we have big A and little b. Inside, little a, big B. And last is the last allele for each trait. The last allele here for A's is little a, and the last one for the B's is little b. So little a and little b. So let's do that here. We'll FOIL big T, big G, little g. First, outside, inside, last. So for the first, it would be big T, big G. Notice that is placed here and here. Outside alleles, big T, little g. Oops. Notice that is placed next, big T and little g. Inside, little t, big G. All right, so you'll see little t, big G. And last, little t, little g. We'll see that here and there. Fix that. All right, so the next step then, after the hard part, is just to do the same thing that we did with the monohybrid cross, where we write down the combination of genes in every single square by writing down what combination where they intersect. So big T, big G, and big T, big G would be in this box. Big T, big G, big T, big G. All right, and so I know this one shows big T, big T, big G, but again, it doesn't matter. That's the same thing as saying big T, big G, big T, big G. The combination of alleles is the same. It's just written in a different order, and it does not matter. It's, again, just a formality. For me, if I were doing this, if I was filling out this Punnett square, I would have wrote big T, big G, big T, big G. Because if I didn't, I would get, like, the letters would start jumbling up in my head. So do whatever way works for you. But that's just the way I would do it. You can do big T, big T, big G, big G. You can do big T, big G, big T, big G, big G. It doesn't matter. It's the same thing. It's just a different order. Order doesn't matter. Next, big T, little g would move into this box, and big T, big G would move into that box. So you would have a total of big T, big G, and big T, little g. And that's how I would have wrote it. However, they wrote it differently. They wrote big T, big G, little g. And again, oh my goodness, this is so annoying. The way I hold my pen, because I don't hold it like a normal person. I say normal person because because most people don't hold their pens the way I do, so it's making me hit those buttons on the side of my pen. So I apologize for that. But anyway, this is saying the same thing. It does not matter the order. All right, so likewise, you would do that with all of the other boxes, all the way down to the last one, little t, little g, little t, little g. All right, and essentially, that would give you a 9:3 to 3:1 phenotypic ratio. So all of these that are shaded in yellow have green potted plants that are tall. In this bluish color, or gray color, I suppose, it looks like a blue-gray to me, they have tall plants with yellow pods. Over here, these three in the purple shaded regions have short plants with green pods. And lastly, the last possibility is a short plant with yellow pods, which is shaded in green. So if you count them up, you'll get 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of these. All that here. I'll just cross them out. There's 9 of those. There's 3 of the grays, 1, 2, 3, 3 of these guys, and 1 of those. So that's why they call it a 9:3 to 3:1 phenotypic ratio. Okay. So the next topic in our textbook is 10.2, and it's how Mendel's laws apply to humans. So one of the most common tools that we use to view family history in regard to different genetic traits are pedigrees. And so you might have heard of a pedigree before in layman's terms. Maybe you've heard about it in genetics, but maybe when discussing animals or something like dog breeds, we want to see their pedigree. And you can create pedigrees for humans as well. And genetic counselors actually do that and help demonstrate and predict how a trait is observed in the lineage of a family, and even help predict the chances of a child being born with a particular phenotype. Now, all of these pedigrees that you can observe, they're going to have some kind of key that will tell you what's what, but what I'm going to list here is some pretty standard stuff. So males are typically squares, females are typically circles, and they can be shaded in different colors that will demonstrate expressing a particular disorder. So typically, if it's shaded, this would be a female that has the disorder. Square would be a male that has the disorder. If there is a horizontal line between a circle and a square, it demonstrates a union. If there is a vertical line down in between them, it will represent children of that union. So let's look at it as an example. Here we have a circle, which would be a female. Here we have a male, because it's a square. And this horizontal line represents a marriage. Or, we say marriage, but it's not like you have to be married to make babies or anything. But this is, again, it's just general. And this is a line of descent demonstrating that they have offspring. This horizontal line demonstrates that they had more than one offspring that are siblings. All right, they had a male child and a female child. And this shows you the generation number here. So this is generation 1. Their offspring would be the generation 2. Now, all of these individuals are unaffected. They do not have the disorder in question. If they did, they would be colored in. Now, some other different things you may see in a key is this symbol, which would mean that they're deceased. This would be twins that are specifically fraternal because we have a male and a female. This is adopted and a miscarriage. Now, not all lines or pedigrees will have all of these, and sometimes pedigrees can utilize different symbols to reflect twins versus adopted and deceased and etc. And again, each pedigree will have its own key. So let's look at some pedigrees for some autosomal disorders. So maybe you're asking yourself right now, what is an autosomal disorder? Well, if you'll recall, we talked about karyotypes. Where's my pen? And there was 22 sets that were shown here of autosomes. And you go back to that if you need to review it. And then the last set over here were sex chromosomes. So an autosomal disorder is a disorder that is caused by an abnormality, a genetic defect of an autosome, one of the first 22 pairs in humans. So more specifically, we'll first look at some autosomal recessive disorders. Autosomal recessive disorders are where a child can be affected even when neither parent is affected. And the heterozygous parents are carriers. So let's do a Punnett square here. If we have two parents who are carriers for a particular genetic defect, which means that they're heterozygotes, they carry the recessive gene. If we were to do a Punnett square, we have a 3-to-1 ratio of our phenotypes, right? 3 squares show the dominant phenotype and 1 shows the recessive phenotype. So the parents here are normal. They do not demonstrate the disorder, but they have a 25% chance of having an offspring that does. And so parents can be tested before having children to see if they're a carrier of this gene and what the likelihood is that they will pass it on to their child. All right, so here is an example of an autosomal recessive pedigree. So we have a female here. She's little a. So she knows she has a disorder. And then, again, this disorder can be many, many different disorders. We'll talk about a few examples. Here's the father. The father is a normal individual for the trait in question, We don't know if he is big A, or big A, little a. All we know phenotypically, he's normal. So, if they have children, this is demonstrating their children, they had 2 children, that are both normal, then they must have the phenotype, or excuse me, genotype of big A, little a. Big A, little a. Because they had to have gotten little a from mom because that's all she could give. And because they're normal, we know that dad must have given his big A. All right, so that's generation number 2. So this individual here, which was a female offspring of the first generation, marries this guy here. He's normal appearance. He appears normal anyway. We don't know his genotype. We just know he must have one big A because he's normal. And their offspring is an unaffected female. So that means then that big A must have came from dad or mom. And they also got a little a, which could have came from dad or mom. We don't know because he could have a little a here. We just don't know. This is why genetic testing is important. All right, so then this person reproduces with this person. And they produce 2 affected individuals and 1 that is not. That we, again, do not know their second allele for. But again, this goes on to show you that affected children can have unaffected parents. Right, just like here in the 3rd generation, this parent 1, parent 2 had children that were affected, even though their parents did not have it. The parents just did not know that they were carriers. So this type of genetic testing is available that allows parents to have a family history of certain autosomal recessive disorders, so they know if they are married to or mating with, whatever you want to call it, a person who also has that gene. The faulty gene, and therefore prevent, or at least know their chances of having offspring with it. Sometimes that affects people's decisions on whether or not they want to have children, for example. Okay, so next, an autosomal dominant disorder. So the previous discussion was over autosomal recessive disorders. In an autosomal dominant disorder, the disorder is present when you only have one dominant gene. So that means regardless if you are big A, or big A, little a, you will have the disorder. So in this case, a child can be unaffected even when parents are heterozygotes and therefore affected. Okay. Then when both parents are unaffected, none of their children will have the condition because there's no dominant gene to pass on. All right, so here's an example of a pedigree that represents an autosomal dominant disorder. So in the first generation, both the male and the female have the disorder. They are heterozygotes. Their children, they have 4 children, 1, 2, 3, 4. Child number 1 is a heterozygote, so they have the disorder. Child number 2, we know they have the disorder, so they have at least a big A. We're not sure about the other letter. We previously would not have known that this was a little a unless we looked at the children. Okay, we know that this individual had to have a little a because they had an unaffected child. They got one little a from mom, so that other little a had to come from dad. So if they didn't have children, if these didn't exist yet, then this would have been big A with a blank space. All right, so next we have 2 unaffected females. And you'll see in the unaffected female's lineage here, once she marries and has children, all of the offspring are unaffected because there's no dominant allele to be passed on. So heterozygotes are affected, homozygous dominants are affected. Affected children are going to have at least one affected parent, which is unlike our previous scenario where we were talking about autosomal dominant, or excuse me, recessive pedigrees. And both males and females are affected with equal frequency. Okay, so let's talk about some examples. An autosomal recessive disorder that relates to the pedigrees we discussed is methemoglobinemia. This is a disorder where there is a lack of enzyme that converts methemoglobin back to hemoglobin. And methemoglobin is a normal byproduct of the recycling of contents in the blood, such as hemoglobin. Once your red blood cells die, they're sequestered by the spleen, and basically your body recycles anything that it can, and then you will urinate or defecate out anything that can't be recycled. So there's an enzyme that enables us to convert methemoglobin back to hemoglobin so we can reuse it for newly created red blood cells, and that enzyme is lacking, and so because of that, there's a decrease in the amount of interaction of hemoglobin with iron and oxygen, and thus makes your skin look blue because blood is red, and that's due to that iron and oxygen and hemoglobin interactions in the blood. So it just makes your extremities, especially the fingertips, look a bluish purplish color rather than a normal, pinkish color as seen here. So this is methemoglobin individual. This is one that does not have that genetic disorder, and basically this is usually a relatively harmless condition. Next, another autosomal recessive disorder is cystic fibrosis. Cystic fibrosis is a very serious genetic defect. It's the most common lethal genetic disorder among Caucasians in the United States. It is a disorder where the chloride channels in the lungs have a defect. So this is representing here a cell that lines the respiratory tract. So here you have the lumen of the respiratory tract, which is the inside of the lungs. And here you have the plasma membrane of a lung cell, and then the inside of the cell. Well, chloride or chloride ions are they need to be able to pass in and out of the plasma membrane appropriately to create the right consistency of mucus. We don't want, there's mucus in our lungs, and that's normal, but we don't want it to be too thick, and we don't want it to be too thin. If it's too thin, it will cause issues. It can cause infections. It will cause pain. If it's too thick, then that could lead to decreased oxygen carrying capacity. It can lead to anemia. It can lead to respiratory problems, and they have trouble breathing, keeping their blood oxygen because the mucus in there is so thick. So this chloride ion defect, or this chloride ion channel defect, causes the mucus in the lungs to be abnormally thick, and therefore reducing the ability of these patients to get oxygen to their blood. It also increases their risk factors when it comes to contracting infections, particularly things like staph is pretty scary for them. Certain staphs, anyway, Staph aureus. Let's see, Pseudomonas aeruginosa is one that's really nasty for them. There's a few others that are also prone to, that people with cystic fibrosis are prone to getting infections with, and it can be deadly for them. So anyway, this is another example of an autosomal recessive disorder due to a genetic defect. Okay, next we have alkaptonuria. So this is an autosomal recessive disorder, and in this disorder we have a lack of functional homogentisate oxygenase. That's a mouthful, but this particular gene is going to produce this enzyme, and that enzyme is normally going to help in the breakdown of products that would normally be excreted into the urine. But because there is a lack of this enzyme, there is a accumulation of what we call homogentistic acid. And that, again, it would have been normally broken down by this enzyme, but because this enzyme is either non-functional or non-existent because the gene for it is messed up, then it's not there to break down homogenistic acid. So it accumulates, and it actually turns the urine black when it's exposed to air. And so these individuals urinate black urine. So That would be definitely very alarming. It also builds up in their tissues, particularly connective tissues, like joints are a really good example. So very uh, interesting to see on the human body. All right, next we have autosomal recessive disorder. This is one of my favorite ones that I like to talk about. It's sickle cell disease. Sickle cell disease is a single base change in the globin gene that affects the amino acid sequence of hemoglobin. So you're going to have to think back here for a minute now, all the way back to like chapter 3. We discussed how DNA would basically tell our cells how to make proteins. And we talked about the levels of protein structure. There's primary, secondary, tertiary, and quaternary. If there is a single base change, that means, let's say we have a base pair sequence of GT, A, T, C, C, G, A. And in reality, this would be, hundreds of thousands, if not millions of base pairs long. Likewise, on this side, you would have C, A, T, A, G, G, C, T, and so on. It would be where we have a single base change. So instead of a T here with an A on the other side, we would have, let's say, a G, which would then make this a C. That single base pair change, we call that a single nucleotide polymorphism, would result in a slightly different protein structure than what would originally be produced by that gene. And this particular gene, if it is a defect of our globin gene, it affects the hemoglobin protein shape. And again, you remember, shape is equivalent to function. So in a normal hemoglobin protein, they are packed into red blood cells, and it makes the red blood cell normal, like that guy. However, if we have a faulty sickle cell hemoglobin protein, then it changes the entire structure of the red blood cell. It makes it sickle shape. It kind of looks like a banana, and it makes the cells sticky as well. And essentially what happens is these sickle cells clog up. They get stuck in little tiny blood vessels. And that means then if you have your red blood cells being stuck in little tiny blood vessels, that there you have poor circulation. You're not getting enough oxygen to your tissues. That leads to anemia. It leads to low resistance to infection. It basically is almost like you're having little mini strokes inside of your entire body. Leads to pain, fatigue. These people that go into sickle cell crisis is definitely not pretty. They come into the hospital, they're, just screaming in pain and have to get a blood transfusion. It's not fun to witness. I saw it at the Mayo Clinic on a few occasions. Okay. Next, Huntington's disease is an autosomal dominant disorder. So that's different, autosomal dominant. Again, that means if you have one allele for the disorder, then it will be seen in your physiology. But this is a progressive degeneration of nervous tissue in the brain. It's a mutation of the Huntington protein. And patients appear normal until middle age. Usually after they have children, they will begin having symptoms. And you have to have a test for the presence of the gene in order to diagnose it. But here you'll see the many numbers of neurons in a normal brain, where if you look over here, this is a biopsy of a person that has Huntington's disease, and you see a large loss of neurons in the brain. All right, 10.3 is beyond Mendel's laws. So we said initially that the Mendelian inheritance patterns don't apply to all traits. Mendelian inheritance patterns assumes that traits are monogenic, and that means that they are controlled by one gene. Mono means one. It assumes also that each trait only has two alleles, one allele that's dominant and one allele that's recessive, and that the dominant allele masks the recessive allele. Now, there are other forms of inheritance patterns. The next one is incomplete dominance. This is where you have a heterozygote that will display an intermediate phenotype. It kind of is like the blending theory that we talked about in the very, very beginning, where if you have a white flower and a red flower, they will reproduce and create pink flowers, because pink is a mix of red and white. So that type of inheritance pattern does exist, but it does not apply to all traits. One example of a human disorder that does follow incomplete dominance heredity patterns is familial hypercholesterolemia. Big word. Let's break it down. Familial means family. So it's genetic. Comes from your parents. Hyper means high. Cholesterol means having to do with your cholesterol. And emia means in the blood. So we have genetic high cholesterol in the blood. That's what familial hypercholesterolemia is. It is an example of a trait that is passed down via incomplete dominance. So a person that has only one mutated allele will have an abnormally high level of cholesterol in their blood. And those that have two mutated alleles will have an even higher level still. So ultimately, here we have a normal artery. It's very clear of debris in there. Here's a cross section of it. You can see the diameter here is very wide, whereas a person that has familial hypercholesteremia will have a very narrow artery because they have lots of fatty buildup and plaque that is building up in the arteries, causing the narrowing of that artery pathway, leading to high blood pressure and an increase in the risk factors of heart disease. All right, so next, this is a chart demonstrating plasma cholesterol for homozygous, heterozygous, and normal individuals. So this is still in regards to familial hypercholesterolemia. You can see we have cholesterol deposits that's so severe it's even, in their extremities, in their hands. You can see some of those bumps here, there, a little bit here, but definitely there. Very large one here and there. So in a normal person, if we were to test their plasma cholesterol, they would have normal levels, which is around 150 to 250 milligrams per deciliter. If you were to test a heterozygote for that trait, they would have a higher plasma cholesterol than normal, around 300 to 550 milligrams of cholesterol per deciliter. And then even more so, if we have a homozygote that has two faulty alleles, they would have a plasma cholesterol concentration of around 600 to 1,000 milligrams per deciliter, which can be very dangerous. So here we see a homozygote. All right, another example of incomplete dominance is hair texture. So wavy hair is an intermediate between curly and straight hair. And hair textures follow an incomplete dominance inheritance pattern. Neither allele is dominant. We have a blend between the two. All right, next, multiple allele traits. So some phenotypes have more than two alleles. Blood groups are a great example. Blood groups can be of three different types, A, B, and O. So I know you're probably thinking, well, I'm A positive or I'm B negative, so there's really more than that, which is true, but we're only talking about the ABO blood group. That plus or minus sign at the end of your blood type is referring to whether or not you have rhesus factor on your red cells. So, and that is a different inheritance pattern. So anyway, we're referring to only the ABO blood group. So there are three possible alleles instead of two. You can have I, big A, I, big B, or little I. A means you have A antigens on your red cells. So here's my little red cell. And an antigen is a protein that can mount an immune response. So there are proteins found on the cell membrane. We've talked about that before. Now you're just knowing they're called antigens because they have the possibility to mount an immune response when given to a person that doesn't have the right blood type. A person that has IB will have B antigens. They're a different shape. Here's my B antigens. And little I means that they don't have A or B antigens. All right, so you have 3 possible alleles, but there's still only 2 allele combination that each person can have. You're just going to have 2 of those 3. So if we have an individual that has alleles I big A, then they're going to have type A blood. If they have I big A little I, they're still going to have type A blood. Because this allele says they have A antigen, this one says that they don't have any. So that means that they will have just A antigens. Another possibility, if they have IA, IB, then they will be type AB. And if they have little I, then that's type O. Type O means you don't have any antigens. All right, so this over here gives you a breakdown of all of the different blood types and the possible genotypes that each blood type can have. If you have type A blood, that means you only have type A antigen. So you can either have this genotype, a combination of 2 IAs, or you can have little I with IA. Type B, you would have a combination of 2 IBs or IB with little I. If you are type AB, then you have to have IA, IB. And in order to be type O, which means you have no antigens, A or B, you would have to have 2 little i's. So both IA and IB are dominant to little I, as seen here. IA masks the expression of having no antigens. IB masks the expression of having no antigens. But IA and IB are co-dominant between each other. You need to know what co-dominant means. It means that they are going to be expressed equally. So if I have type AB blood, I will have both A and B antigens. All right, and so this shows you inheritance of ABO blood types, or an example of it. Here we have parents that are IB, little I, that means they are type B blood. And we have IA, little I, which means they are type A blood. More specifically, we could also say they have type BO blood and AO, which is more specific. But, of course, the B and the A are dominant over the little I, which is O. Okay, so if these 2 individuals had offspring, we would line up the potential gametes, which for the female here would be IA and little I. So IA, little I. Likewise, IB, little I. And we would fill in the chart just like we did the rest of the Punnett squares. And we have a 1:1:1:1 ratio of our phenotypes. They could be IA, IB, which is type AB blood. There's a 25% chance of that. They could have B blood. They could have A blood, or they could have O blood. 25% chance of each. All right, next, polygenic inheritance. Polygenic inheritance means that it is a trait that is governed by at least 2 or more sets of alleles, 2 or more genes. And as a matter of fact, most traits are polygenic. So a lot of times we'll talk about things like eye color and hair color as if they're monogenic, but that's for simplicity's sake. In reality, hair color, eye color, height is a polygenic trait, which means that it is governed by multiple genes. Each dominant allele has a quantitative effect on phenotypes, and the effects are additive, and as a result, we will see a continuous variation, which leads to a bell curve shape if we observe the population, and you'll see that on the next slide. Polygenic traits are also tend to be subject to environmental factors. So traits that are subject to environmental factors are called multifactorial traits. So even hair color is a polygenic inheritance trait controlled by many genes, and it is also multifactorial because it's not just about genes. It's controlled by the environment. For example, if you go to the pool a lot and you're blonde, it might turn your hair a little bit of a green shade. When I was born, my hair was really blonde, but as I got older, my hair turned pretty dark brown. So part of that is genetic and part of that is environment. Some other examples of multifactorial traits that also has environmental effects are fetuses that are born with cleft lip and palate. Diabetes has a genetic component, but it also has to do with how you eat, which is an environmental factor. Schizophrenia has genetic and environmental factors. Allergies, cancer. You can be genetically predispositioned for skin cancer and never get it as long as you protect yourself from the sun. And again, these polygenic traits, which tend to be multifactorial, have a continuous variation, which is demonstrated by a bell curve in a population. So when I say continuous variation, we'll talk about height as an example. You're not either tall or short, right? That's not how continuous traits work. Some people are 62 inches tall, or 63 inches tall, or 63 and a half inches tall, or 63.75 inches tall, or 68 inches tall. And y'all see the point that I'm trying to make. It's not it's not this or that. It's not tall or short. There is a wide array or a range of different heights that people can be. And if we were to look at a population of individuals, you'll see that most individuals are approximately this height, around 68 inches. And then as we move away from the average, we see less and less people of those within the range of that particular height that it associates with. So we have a little bit less people that are 70 inches tall. We have a little bit less people that are 66 inches tall relative to the average. We have even less people who are 64 inches tall. We have even less people who are 72 inches tall, and very few people are 74 inches tall. And very few people are 62 inches tall, and so on and so forth. So this is why we call it a bell curve. Because if we make a graph, it literally looks like a bell. Another great example of a continuous variation in a polygenic and multifactorial trait is skin complexion. So skin complexion, the color of your skin, is in response to ultraviolet radiation as well as your genetics. When our skin is exposed to UV radiation, melanin is produced. Melanin is a pigment. And the more melanin our skin produces, the more, the darker complexion that we have. And in addition to that, human production of melanin in the skin increases closer and closer to the equator as to help protect our skin from the sun. Because if you were to look at the shape of the earth, it's round, right? So, here's the sun. The sun is closest to the earth and spends most of the time closest to the earth at the equator. All right, so this is where the strongest UV rays are. And that's also why, if you look at the map of the world, where the equator is, it's associated with people that have genetically darker skin than those that live in towards the poles of the earth. All right, and so this shows us here the number of dominant alleles that we have relative to what the skin complexion would look like. So we see over here we have very, very fair skin with no dominant alleles. And over here we see 6 dominant alleles, 1, 2, 3, 4, 5, 6, and very dark complexions. And then as we progress towards the center, we see a number of dominant alleles that is 3, which is most common. So most skin complexions across the world fall here, and we have less and less on either side as we go. All right, next, some other examples of variations of inheritance patterns is that some genes interact with each other. So multiple pigments, for example, are involved in determining eye color. That's why we don't have just brown or blue eyes, which is how we would discuss it if it fell in Mendelian genetics patterns, but they don't. You even have green eyes. Some people have very icy blue eyes. Some people have gray eyes. Some people have hazel looking eyes. So and that's because there are multiple genes that control eye color that interact with one another to determine the final color. And I believe this is our last concept. Oh, nope. But the last concept in this section is pleiotropy. And this is where single genes have more than one effect. So sometimes having a faulty gene can have mass effects on the human body. And a great example of that is Marfan syndrome. It is due to the production of abnormal connective tissue and creates lots of issues systemically throughout the body. So, for example, the skeleton, which is, of course, made up of bones, which is a type of connective tissue. It will result in chest wall deformities. They're usually very tall. They have long, thin fingers, arms, legs. They may have scoliosis, which is a curvature, a defect of the spine. Flat feet, instead of having an arch like most people do. They have heart and blood vessel abnormalities because blood vessels, or blood anyway, is a type of connective tissue, and connective tissue is found in the blood vessels. So they may have mitral valve prolapses, enlargement of the aorta, which is a very, very important aorta or artery in the body that can lead to an aneurysm, and that is very, very deadly. The eyes, they can have lens dislocation. So we have lenses within our eyeballs that help gather light and focus it in such a way that we can see. And because that's that lens can be dislocated, it causes eye problems. They can be very, very, very nearsighted, where they can't see from very far away. They can also have lung issues. They're prone to having collapsed lungs, because there is lots of connective tissue, like elastic tissue, that is found within those tissues in the interstitial spaces of cells. They can also have skin defects. All right, so next, and I believe this is the last concept in this chapter. This is a really long chapter, but is linkage. So two traits that are on the same chromosome are said to be linked genes. If there are two traits that are found on the same chromosome, then they do not segregate independently. So remember, we discussed how our chromosomes separate into cells during meiosis independently of one another. So, however, if we have two genes that are on the same chromosome, then those two genes will be linked and most likely stay together during the process of meiosis and creation of sperm and egg. Okay. Now, we also talked about recombination in meiosis 1, specifically prophase 1. Remember, that's where we have a maternal and a paternal portion of a chromosome break off and switch places. But linked alleles will stay together during that process. Lastly, 10.4 is sex-linked inheritance. So females are genetically XX. They contain 2 X chromosomes. All eggs after meiosis will have only 1 X chromosome. Males are XY. So sperm contain either an X or a Y. The Y chromosome carries an SRY gene, which determines maleness. That's why males are considered genetically XY, and females, because they don't have a Y, are considered female. The X chromosome is much larger and carries more genes, and so there's more examples of X-linked chromosome disorders than there are Y-linked. So let's look at this picture here. It demonstrates how sex determination works in humans. So in a female, we have 44 autosomes, and we have a set of sex chromosomes. They will be XX for a total of 46 chromosomes. In a male, we have XY. So after meiosis occurs, this egg will have 22 chromosomes plus an X, because that's all she can give to the egg is X. That's all she has. She didn't have a Y. So an egg will always have an X chromosome for a total of 23 chromosomes total. 22 plus the X chromosome makes 23. Now in sperm, they can have 22 autosomes plus an X, or they can have 22 autosomes plus a Y. And depending on which sperm fuses with this egg will determine sex determination. So if this sperm, the one with an X, fuses with this egg, it will produce a female child. If this sperm, which has a Y chromosome, fuses with this egg, we will have a male child. So you are a female or you are a male because of your dad. It had nothing to do with your mom. Sex determination is determined by the sperm. All right, so now let's relate that to sex-linked alleles. Not all alleles or not all genes are located on chromosomes, which are non-sex chromosomes. Some of them are specifically on the X or Y chromosome. And again, because the X chromosome is larger, there's more examples of X-linked traits. A great example is seen in fruit flies. We'll talk about some human examples, but fruit flies provide a great model for sex-linked inheritance patterns. Eye color in fruit flies, which is also called Drosophila melanogaster, that's their scientific name, is determined by a gene on the X chromosome. So it's an X-linked trait. So when a red-eyed female mates with a mutant white-eyed male, all the offspring have red eyes. In the F2 generation, there's a 3-to-1 ratio that was found, but all of the white-eyed flies were males. The Y chromosome does not carry alleles for X-linked traits, so everything's dependent on the X from the female parent and not the male. So let's look at the X-linked inheritance Punnett square here, because that'll really help us kind of understand this. So here we have the parents. This one is male because it has a Y. This one is a female because it doesn't. And the XB, capital B here, means normal vision, red eyes. XB, again, normal red eyes. And X little b is white eyes, if we were discussing it in terms of these Drosophila, but it also relates to human disorders like color blindness. So that stands for a colorblind allele. They both have normal vision, these parents, because they both have at least one dominant allele. Now let's look at the offspring. So here we have the gametes of the parents. This would be from the female parent, XB that's little. And then over here we have the gametes of the sperm, XB and Y. So then we would fill in the Punnett square just like we would for any other Punnett square. And as a result, you see we have a homozygous dominant individual that is female. We have a heterozygous female. We have a male that has normal vision. We call this hemizygous because they only have one allele instead of two. And then we have a colorblind male. So this cross appearance resulted in a 3-to-1 phenotypic ratio of colorblindness. All right, so in a pedigree for sex-linked disorders, what you will see is that sons, male children, inherit the trait from their mothers. The son's X comes from the mother, because if they're a male, then the father had to have donated a Y. So more males than females have X-linked disorders because the allele on the X is always expressed in males because they only have one X chromosome. So for example, if I had a female, she has two X chromosomes, and this gene is mutated, well, that's okay because we have an extra copy right here that's good and is fine. So if this was a colorblind allele and a normal allele, well, this person will be able to see in color. She's a female, she has two X chromosomes. However, if this same individual is a male, XY, and this color blindness gene is faulty, then this male will be colorblind because they only have one X chromosome. And so if the gene on it for color blindness is messed up or defective, then they don't have that second X chromosome to counter the faulty allele. So that is why males tend to have X-linked disorders more frequently than females do. Females who have the condition inherited the mutant allele from both their mother and father. So if we have a father who is colorblind and a female that is colorblind or is a heterozygote and happens to donate the faulty gene to their offspring that is female, then that female will be colorblind. But that's only if they have both parents that carry that allele. All right, and lastly, condition appears to pass from grandfather to grandson. Here is a X-linked recessive pedigree for colorblindness. So here we have the grandfather. We have his offspring, which is a male that is unaffected and a female that is unaffected. The female here has a husband that is not affected, but she carries the faulty allele, X little b. They have 4 children, one of which is a male that has the disorder because they got the Y chromosome from dad, and just by happenstance, they got the X little b from mom, which means they are affected. That's why it goes from grandfather to grandson. Now, of course, that's not always the case. Here we have a male child that they had, which is unaffected, an unaffected son of the grandparents. And he marries a female who is colorblind. Well, that offspring is also colorblind because they got the XB, little b, the defect from mom, and they got a Y chromosome from dad. Now, if they had gotten X big B from dad instead, and it was a female child, then they would not have had the disorder. So again, that just represents how it passes from grandfather to grandson typically, and how males are affected more so than females. All right, there's only a few traits that are X-linked dominant. Excuse me. Up until this point, we talked about X-linked recessive traits. There's very, very few that are X-linked dominant, and if that's the case, daughters are affected. Daughters of affected males have the condition as well. So affected females can pass the condition to daughters and sons. It just depends on which X is inherited. Now, Y-linked disorders, there's also very few because, again, the Y chromosome is so small. And it is present only in males, obviously, because only men have the Y chromosome. And if there is a defect on the Y chromosome, it is passed to all sons but not daughters. All right, so just to list a few examples of these X-linked recessive disorders, we already mentioned color blindness. It's about 8% of the Caucasian men have red-green color blindness. They're the ones that are most commonly affected by color blindness, and this is the specific type, red-green color blindness. It affects the cones, and they're called. There's 2 types of cells that are in the. This is the eyeball. All right, and then at the very back of it, we have the retina, and the retina has cells that line it that are called cones and rods. The cones are utilized for color vision, and there's different types of cones that are used for viewing different colors. And so the red-green color blindness will affect the cones that are used to see those colors. Secondly, we have Duchenne muscular dystrophy. This is where we have the absence of the protein dystrophin, which causes a wasting away or atrophy of the muscles. There is some hopeful therapies for this disorder, where immature muscle cells can be injected into the muscles and theoretically, hopefully, grow and be able to be utilized by the patient, but it's pretty new. This is a picture showing you the difference between a biopsy of a leg tissue that for specifically muscular tissues in the leg of a person that is normal versus one that has muscular dystrophy. So in a normal person, you will see there is relatively little space, and it's in the light pink. That light pink is representative of the connective tissue that encapsulates muscle cells and fibrils. The dark purple is the nuclei of muscle cells. And then the muscle filaments that actually contract and are able to generate force are in the darker pink. So in a muscular dystrophy patient, you see a lot more fibrous tissue. That's the light pink. It's connective tissue. And then much less than of the normal healthy muscle tissue. And so essentially it's a progressive disorder where the muscle tissue just gets weaker and weaker and weaker and degenerates over time, to the point where they'll eventually be wheelchair bound and eventually can lead to death over time. So, but again, it is a X-linked recessive disorder. All right, so that is chapter 10. I know this video was kind of long, but luckily we only have one chapter to cover this week. I hope y'all enjoyed the narration.